In this video session, we'll be discussing about transport of macromolecules. As they are large molecules or large particles, they cannot easily pass through the cell. So basically, there are two ways through which macromolecules can enter into the cell. Ways are endocytosis and exocytosis. So let us discuss about each one of them. Some macromolecules cannot pass through the cell membrane either by active or passive transport mechanism. Such substances are transported into the cell by endocytosis. So it won't be wrong if we define endocytosis as a transport mechanism by which macromolecules enter into the cell. If we talk about types of endocytosis, then there are three types of endocytosis, and they are Pinocytosis, Phagocytosis and Receptor-Mediated Endocytosis. So first of all, let us talk about Pinocytosis. Pinocytosis is a process by which macromolecules like bacteria, antigen, etc. are taken into the cell. To be more clear, let us see the mechanism of Pinocytosis. At the very first stage of Pinocytosis, the macromolecules in the form of droplets or fluids bind to outer surface of the cell membrane. And after that, the cell membrane invaginates around the droplets of macromolecules, and the droplets are engulfed by cell membrane. The engulfed droplets are converted into vesicles and vacuoles and are collectively known as endosomes. Now the endosomes travels into the interior portion of the cell. The primary lysosome, which is already present in the cytoplasm of the cell, now fuses with the endosome and forms secondary lysosome. The hydrolytic enzymes present in secondary lysosome are activated, resulting in digestion and degeneration of the endosomal content. In this way, the macromolecules are consumed in the form of droplets or fluids by the cell, so it is also known as cell drinking process. Second type of endocytosis is phagocytosis. Phagocytosis is the process by which particles which are larger than macromolecules are engulfed into the cell. That's why it is also known as cell-eating process. Larger bacteria, larger antigens and other foreign bodies are taken inside the cell by the means of phagocytosis. And only few cells inside the body like neutrophils, monocytes and other tissues, macrophages, shows phagocytosis. Now let us shortly discuss about the mechanism of phagocytosis. When bacteria or foreign bodies try to enter into the cell, at the very first place, the phagocytic cell sends cytoplasmic extension, which is also known as pseudopodium, around the bacteria or foreign body. Then these particles are engulfed and are converted into endosome-like vacuoles, and vacuole is very large and is usually called phagosome. Now phagosome travels inside the cell. The primary lysosome fuses with the phagosome and forms secondary lysosome. Hydrolytic enzymes present in the secondary lysosome are activated, resulting in digestion and degradation of phagosomal content. In this way, the cell eats larger bacteria, larger antigens and other foreign bodies. That's why it is also known as cell-eating process. And the third type of endocytosis is the receptor-mediated endocytosis. So the receptor-mediated endocytosis is the transport of macromolecules with the help of receptor protein. The cell membrane has some pits which contains receptor protein called clathrin. So together with the receptor protein, each pit is called receptor-coated pit. These receptor-coated pits are involved in the receptor-mediated endocytosis. Now let us shortly discuss about the mechanism of receptor-mediated endocytosis. Receptor-mediated endocytosis is induced by substances like ligands. Ligand molecules approach the cell and bind to receptors in the coated pits and forms ligand-receptor complex. Now the ligand-receptor complex gets aggregated in the coated pits. The pit is detached from the cell membrane and becomes coated vesicles. And those coated vesicles, they form endosomes. Now endosome, they travel into the interior portion of the cell. Primary lysosome, which is present in the cytoplasm, fuses with the endosome and forms secondary lysosome. And the hydrolytic enzymes which are present in the secondary lysosomes are then activated, resulting in release of ligand into the cytoplasm. After that, the receptor may move to new pit of the cell membrane and continue the cycle. We talked so many things about endocytosis. We even talked about the types of endocytosis. Now let's talk about exocytosis and its mechanism. So endocytosis was all about how the macromolecules enters inside the cell, but exocytosis is related with how the macromolecules are expelled out from the cell. In this process of exocytosis, the substance are expelled out from the cell without passing through the cell membrane, and this is the reverse process of endocytosis. So for now, let's talk about the mechanism of exocytosis. Exocytosis is involved in the release of secretory substance from the cells. The secretory substance from the cells are stored in the form of secretory vesicles in the cytoplasm. When required, the vesicles approach to the cell membrane and gets fused with the cell membrane, and later on the content of the vesicles are released out of the cell. Now in very short, let's talk about the role of calcium ion in exocytosis. Calcium ions plays an important role during the release of some secretory substance such as neurotransmitters. The calcium ions enters into the cell and cause exocytosis. However, the exact mechanism of exocytosis is not clear.